and 3's. It's fun to play with some bDNA. It's got a boatload of GCTA. With those hydrogen B's and right-hand helices, anti-parallel 5's and 3's. That's enough. Okay. Okay. By the way, if you've gone to my website, for a long time I had the recordings of the songs off. This past weekend I actually put them all back on there. So if you'd like to download real recordings, not me, but actual professional recordings of them, they're all there on the website now. So, and you can give them away to your friends. I don't try to make money on these. They're out there for you to use. Okay. Now, so we've gone through a fairly involved process right there. Okay? Translation, it turns out, is a very important mechanism to understand, because an understanding of translation allows us to manipulate it. And the main manipulations that we do of translation are in controlling bacteria. Okay? So it turns out that there's several antibiotics that specifically inhibit steps in bacterial translation that are different from what we have. Steps in bacterial translation that are different from what we have. And perhaps steps isn't quite the right word to use, because the steps are basically the same. Rather, they have different structures. Bacteria use different proteins than we do in their ribosomes, and so as a consequence have slightly different structures within them. So if you can design an antibiotic that binds to a structure in a bacterial ribosome but doesn't affect yours, you've just found a way to kill that bacterium. So several antibiotics that I'll briefly mention here work by exactly that mechanism. Okay? Exactly that mechanism. One of these is puromycin. Okay? Puromycin, okay, has a structure that looks like this. Right? It looks kind of like a tRNA, and it kind of looks like it's got something attached to it. And it turns out that puromycin will actually bind in the A site of a bacterial ribosome. It will not bind in the A site of your ribosome. Consequently, puromycin will stop translation, because when it gets there, the ribosome can't attach everything and continue the process. So puromycin is mucking with elongation. It's mucking with bacterial elongation. Okay? It stays bound. It just gums everything up. And so the ribosome can't do anything, and everything just sort of stays stuck. So again, when you screw up translation for an organism, you're going to kill that organism, or for that cell, you're going to kill that cell, because it's not going to be able to go on and do its thing. Is there a question over here? Okay. Okay. All right. That's a list of components. No, you're not going to memorize that. Okay. And polysomes. What's a polysome? Okay. Actually, let's not talk about that either. We'll just finish that. Okay. Let's move on to eukaryotes. All right. So eukaryotes. Now, as I said, the process of translation in eukaryotes is very similar to that in prokaryotes. The proteins have different names, and I don't think it's really going to teach you anything if we go and we memorize all those names. Right? You had enough names in the prokaryotic system to understand that. I will point out a couple differences, and they relate to S's. Okay? They're very simple. All right? In the bacterial system, we had a 30S and a 50S. They made a 70S, right? In eukaryotes, we have a 40S and a 60S, and they add up to an 80S.
Those are all for the same functions of each.
And they have the same basic functions. The eukaryotic system has different ribosomal RNAs with different S numbers, and for our purposes, I think I've just decided not to just give you those. So we have an equivalent to the 23S that makes peptide bonds. We have an equivalent to the 16S that the bacteria have. I'm going to point out to you that we don't have Shine-Dogarno sequences, and I'll explain why that is the case in a minute. But the one that we have is equivalent to that ribosomal RNA, the 16S ribosomal RNA of bacteria. Okay? And in addition, the differences that we have relate more to the structures of the messenger RNAs. Last time, you'll remember I told you that eukaryotic messenger RNAs have two significant modifications, one to the 5' end and one to the 3' end. Anybody remember what was at the 5' end? The cap. Very good. Thank you. I was hoping to just squeeze that out of you, so thank you, Matt. Okay? The cap. At the 3' end, we have the poly-A sequence, right? It turns out that both of these they do play a role in translation in eukaryotes. So this is one difference that we see in eukaryotes compared to prokaryotes. The cap and the poly-A have roles in the process of translation. Okay, there's a lot of S's there. There's a lot of things there, and pre-initiation complexes and blah, 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 blah. And you see all these different names that are there. The overall process, for our purposes, is the same. Okay? One of the differences that we see is something called RNA looping. All right? RNA looping. Now, remember that in bacteria we have operons. What's an operon? An mRNA that codes for more than one protein. All right? We don't have that in eukaryotes. In eukaryotes, you have one messenger RNA, one protein. Okay? Consequently, if you think about this, when eukaryotic translation is occurring, if we think about, let's say we start translation down here, we make it go all the way around and get up here, and it hits a stop codon. If we've got a little loop like we have here, the ribosome can kind of easily make its way back over here and do this process over and over and over and over. The loop helps translation to occur more frequently. In prokaryotes, we have multiple genes, so the loop doesn't make any sense. We finish one gene, we move on to the next gene, to the next gene, to the next gene. So prokaryotes do not do looping, we do looping. And the looping, as you can see, is facilitated by, boy, there's a lot of proteins with a lot of names there. This is where the cap and the poly-A tail play roles. They're recognized by different proteins that help to make the loop. So the cap is recognized by this protein here. The poly-A tail is recognized by this protein. I'm not asking you to memorize the names. But now this whole complex is held together so that we can have initiation, elongation, termination, and then go through the whole process again. Okay? If you give a eukaryotic cell a messenger RNA that lacks the cap or that lacks the poly-A, it will not be translated. So eukaryotes need those structures in order for translation to occur. Okay. Elongation, termination, blah, blah, blah. All right. That is translation. We did that a little faster. Oh, I've run out of time. Sorry. Since we're going to have a review session, I guess that's okay to run out of time today. So any questions on that before I take a brief break?
So eukaryotes don't have operons?
Eukaryotes do not have operons. Okay, let's take about a 2-minute break. If you want to hit the bathroom or whatever, I'll give the camera a break, and then we can get started on a review session.